Okay, welcome everyone. I'm happy to see a lot of familiar names and faces, and this is the research interest group Bridging Disciplines to Find Solutions for Osteoarthritis. And so welcome on behalf of myself, Rachel Miller at Rush University, and also my co-coordinator, Tim Griffin at the OMRF. So we have an exciting program here today, and before we get started, I just want to mention a few housekeeping items on behalf of the ORS. So number one, thank you to the ORS and to all of the ORS staff who've made this session possible. And just please keep in mind that the session will be recorded, including the chat service. And so in a few days, it should be available for watching after the fact. If you could, as participants, please keep your video off and yourselves muted during the large session in order to preserve bandwidth. And then we'll be doing breakout room sessions where then you'll be able to do smaller group interactions with people. Recording or screenshots of any kind is not appropriate without prior approval by the ORS and by the author or speaker. You can use the chat box to submit questions to us at any time. And we'll also be having a poll towards the end of the research interest group that you can take part of. And finally, if you enjoy this session, we invite you to become members of the ORS, and you're welcome to visit the Global Networking Lounge after this talk or any time during the meeting in order to interact with other participants and also the ORS staff members. Okay, so let me share my screen here. And so, as I mentioned, this session is Bridging Disciplines to Find Solutions for Osteoarthritis. And so the concept for this research interest group was thought up by actually Tom Andriaki and Richard Loser a few years ago. And so the idea is to bring together different researchers in the area of osteoarthritis, but also related disciplines in order to brainstorm and come up with future research topics moving forward. And so we all now recognize that osteoarthritis is a disease of the whole joint, and you can image and visualize these changes within the joint. This is just the example of the knee joint, where you can see joint space narrowing by a radiograph, soft tissue changes by MRI, and macroscopic loss of cartilage. And so while all of these changes are happening, it's now recognized that the main symptom that patients experience with these changes is pain. And so that will be the focus of the talks this year, trying to examine from different angles how we can study the relationship between these structural changes in osteoarthritis and pain. And so I just wanted to quickly highlight a lot of the people that are participants here in this research interest group also have a lot of nice programming as part of the meeting. So please take advantage of watching these talks and interacting with these people. Let's see. So just as an agenda for the research interest group, we're going to have 3 short talks, 15 minutes each. Then we'll be able to use the breakout room function in order to choose different speakers to interact with. That'll be for 20 minutes. Then we'll all come back to the main room and hopefully have a larger group discussion for the remaining 20 minutes, where we'll also give a little summaries of what was happening within the different breakout rooms. So in case you're new to the breakout rooms, at the bottom of your Zoom screen, when it's time, there will be a breakout room button that will appear. Then you can see the different options, and you have to actually hover your mouse over these numbers next to each label and then click join to join the different breakout rooms. Okay, so with that, I'm going to ask Carla to start loading her slides. And in the meantime, I will give a brief introduction. So Dr. Carla Scanzello is the section chief of rheumatology and is the co-director of the multidisciplinary OA clinic at the VA Medical Center in Philadelphia. She's also an assistant professor of medicine at the University of Pennsylvania School of Medicine. Dr. Scanzello received her medical and graduate degrees from Temple in Philadelphia, and she completed a residency training at the New York Presbyterian Hospital in the Cornell Medical Center in New York. And she did her rheumatology fellowship at the Hospital for Special Surgery, also in New York. She then moved to Chicago, where she was my colleague at Rush for 5 years before moving back to Philadelphia and joining the faculty there. She's an active member of the ACR, the ORS and OARSI, and her research is funded by the National Institutes of Health, as well as the VA. Okay, Carla, I'm happy to invite you to share your work with us.
Great, thank you. Can everybody see my slides and hear me? Okay. Well, thanks so much to Rachel and Tim for inviting me to give the kickoff talk. I am looking forward to not only talking to you about what we know and what we don't know about the relationship between inflammation and pain and osteoarthritis, but also to the discussion afterwards. I have no disclosures. So as a medical student and then onward through my clinical training as a resident, I learned that arthritis pain is often separated into two main categories, mechanical pain or inflammatory pain. Mechanical pain, we were taught, is worse with movement, improved with rest, and associated with minimal stiffness, while inflammatory pain is improved with movement, worse with inactivity, and associated with significant stiffness. And that this provides a useful framework for understanding and trying to figure out what's going on in our patients who have arthritic pain to help us determine whether this is likely osteoarthritis, characterized by mechanical pain, or rheumatoid arthritis, or one of our other inflammatory arthritides that we treat in rheumatology. So simple, right? So I think this audience appreciates the fact that pain and osteoarthritis is actually quite complex. And I'm not going to go into details here because our second speaker, Dr. King, is going to talk a little bit more about this. But suffice it to say that at least one level of complexity is the fact that characteristics of OA pain change with progression, whereas pain might start off as intermittent and largely related with movement or weight bearing. It can progress in many patients to become more constant and persistent. It has a lot of different qualities and different phenotypes and different trajectories and different populations. But one of the mechanisms that has been an area of research in this transition from intermittent to constant and persistent pain is potentially the development of peripheral and central sensitization. So signs that the pain transmitting system from our joint to our central nervous system is being remodeled, just as the joint is in OA, is quite substantial, leading to heightened sensitization of the system to transmit pain. And in terms of relationship with inflammation, it's been known for some time that peripheral and central sensitization can be facilitated by inflammatory mediators. And this is taken from a review on peripheral sensitization just from 2014, showing you the broad range of inflammatory mediators that can bind to their cognate receptors expressed on nociceptive nerve terminals in a variety of tissue contexts and stimulate this process of sensitization. So when I think about inflammation in OA, I think it's useful to think of at least two different definitions or two different aspects of inflammation. I say at least because I'm sure there are more. I know there are more, but I just present this dichotomy for discussion. That mechanoinflammation, which which Tanya Vincent's work has contributed to, and she has reviewed quite a lot, describes the process by which mechanical input on mechanically sensitive cells like chondrocytes in the joint can lead to inflammatory signaling cascades that lead to the catabolic phenotype. And this is obviously has a lot of evidential support that this is a key aspect of inflammation in osteoarthritis and important to talk about. But as somebody who trained in rheumatology, we are used to thinking about inflammation from the aspect of the immune system and cellular infiltration of leukocytes from the immune system into sites of inflammation in various tissue contexts. And so there's plenty of evidence that inflammation, this type of cellular infiltration is occurring in OA, particularly in the vascularized tissues such as the synovium. In terms of OA pathology, these two aspects of inflammation set up a very complex crosstalk between tissues and cells within the joint that can contribute to progressive pathology. But these aspects of inflammation are both likely relevant to pain as well, in that they can both lead to production of pain mediators such as the prostaglandins, pain sensitizers such as various chemokines and NGF, and lead to development of synovitis, which has been at the imaging level associated with both pain severity and pain sensitization in OA patients. That these infiltrating leukocytes may play roles in OA pathology and OA pain has some evidence, although this, I think that this is an area that the field really needs to grow. But there's multiple types that might be relevant here, multiple cell types. So Shen and colleagues in 2011 provided evidence that the lymphocytic infiltrate might be relevant, in that mice deficient in CD4 helper cells showed some protection from synovitis and cartilage erosion in an ACL transection model. More recently, Christopher Little's group showed in the DMM model that mice who were treated with either an HA derivative or mesenchymal stem cells injected into the joint showed reduction in mechanical allodynia after treatment, and that this reduction was temporally correlated with reductions in synovial lymphocytes, suggesting a potential role in OA pain as well, but it needs to be more explored. Other cell types, other resonant cell types within the joint are relevant here as well. So Bill Robinson's group in 2019 provided evidence that mast cells, which reside in the joint, they're not necessarily increased in the setting of OA in this model, but they are activated, and that targeting them either through genetically or pharmacologically attenuates OA pathology. Relevance to pain is not clear yet. But the bulk of the work in terms of leukocyte subsets in OA has been on synovial macrophages. And that has produced a pretty complex picture. So in the collagenase-induced arthritis model, depletion of macrophages from the synovial space can promote OA, particularly osteophytosis. But in other models, here a high-fat diet DMM model, and more recently from Steve Olson's group, intra-articular fracture model, depletion of macrophages, or macrophages might play an important role in protecting from OA pathology. And in the 2017 paper from Farshguilak's group, where they used the mafia mice to conditionally deplete macrophages before induction of osteoarthritis in the high-fat diet DMM model, they showed that synovitis and pathology was actually increased. And in particular, the synovitis was characterized by the influx of other cell types, particularly lymphocytes, suggesting that synovial macrophages, at least subsets, may be involved in protecting from activation of other aspects of the immune response, particularly the adaptive immune response. So our group has been thinking about how inflammation might be relevant here in multiple, and how it might be relevant to multiple cellular contexts. And so we've been thinking about the fundamental question, how can inflammation be triggered by joint tissue injury? To do this, we have been looking at the pattern recognition receptor systems of the innate immune response, in particular, the toll-like receptors and their ligands. Now, toll-like receptors have been shown to be crucially important to the initiation of inflammation in pathogenic contexts, but also in sterile injury. And these receptors can be triggered by endogenous products termed damage-associated molecular patterns, or DAMPs. A number of TLR ligands have been identified in the OA joint. We'll get to that in a moment. And blockade or deficiency of individual TLRs in animal models, though, has been relatively disappointing, with most studies looking primarily at pathology and not necessarily pain-related outcomes, with one notable exception, and that is work from Mary Beth Humphrey's group, where they showed that TLR4 knockouts were relatively protected from development of OA in an obesity model. One of the factors that might be contributing to a complex response with regards to these receptors are not only the fact that there's a lot of redundancy in terms of multiple receptors on the cell surface in the endosomal compartment, but the breadth of types of DAMPs that have been described in OA. These include LPS, soluble products produced by stressed or dying cells, such as HMGB1 and the S100 family of proteins, and products of extracellular matrix catabolism. And in particular, one matrix product, a 30-tumor peptide produced from aggrecan that you'll hear about again in just a moment, as well as self-nucleic acids. And there was a nice poster presented today in one of the sessions about mitochondrial DNA and OA. And so all of these ligands can bind to a number of different receptors, and we don't understand the complexity in terms of whether one of these is most important in OA or not. In addition to that complexity, there's a number of TLR co-receptors that can influence the strength of signaling through the TLRs. These include LBP, which assists LPS to binding to TLR4, as well as a molecule that's caught our attention, and that's CD14. CD14 can influence signaling through multiple TLR receptors, and it's active as both a soluble and a membrane-bound molecule, both at the cell surface and in the endosomal compartment. So we first got interest in this particular molecule, which can influence TLR signaling, when we identified it in synovial fluids from patients with early and advanced OA. And we showed that levels were increased compared to asymptomatic controls, and more importantly, they were increased locally in the synovial fluid, suggesting local production. Others, Virginia Krause's group, went on to show that CD14 has biomarker potential, and that's associated with pain severity in patients. We've been investigating whether CD14 deficiency can affect pain and pathology in the DMM model, and this is work done by a former postdoc in the lab, Nisha Sambamurthy. And we've been using, to look at movement-associated pain, we've been using the ORS system, which are vibration-sensitive platforms, which allow us to look at mouse spontaneous activities. And in CD14, we followed mice for 16 weeks, measuring these outcomes at 4-week intervals. And what we find is that CD14 deficient mice, in addition to having some protection from pathology, also shows some protection from movement associated pain measured, and particularly in climbing activity. So that TLRs might be a link between cartilage damage, inflammation, and pain in OA has been suggested by a line of questioning. So Amanda Fossang's group first showed that one of these DAMPs from matrix degradation to aggrecan 30 tumor can stimulate chondrocytes via TLR2 mediated mechanism promoting catabolism. And then Rachel Miller and Anne-Marie Malfate went on to show that there might be a direct effect on pain sensitization, that DRG neurons express TLR2, and that mice who cannot produce the aggrecan 30 tumor are protected from pain sensitization in the murine DMM model. So this work, I think, suggests a model of neuroimmune crosstalk in playing an important role in OA pain. I do think it starts with mechanics and mechanoinflammation in the large majority of patients. And this mechanoinflammation promotes matrix catabolism. And then DAMPs produced in that setting can then feed back and perpetuate the inflammatory response at the level of the chondrocyte. Although I also want to mention that there's some data out there suggesting that some TLR signaling in chondrocytes may be protective of homeostasis as well. So it's complex. But these same DAMPs produced in the setting of inflammation and catabolism can influence the activity of synovial cells, particularly synovial macrophages, but also potentially fibroblasts via TLR-mediated mechanisms, allowing these cells to contribute to the proteolytic milieu in the joint, promote production of chemokines that can stimulate and perpetuate synovial inflammation, and can produce a number of soluble mediators that can sensitize nociceptive neurons who have nerve terminals within the joint tissues. But of course, the chondrocytes themselves can produce those same pain sensitizers in their inflammatory activity. And I think one of the things that we need to understand in the setting of OA is what are the predominant cell sources and what are the predominant cell targets that are relevant here. So our second talk, you're going to hear a lot about how pain can be measured to help us understand this response better in animal models from Dr. King. And Dr. Franz is going to discuss the influence of mechanics and whether we can modulate mechanics to have a positive influence on the joint and break this cycle of inflammation that's acting at multiple cellular levels. So the take home is that pain is extremely complex. We're talking about mechanics and inflammation, not mechanics or inflammation. And that the pathways of innate immunity may provide a link between mechanics, inflammation, and pain. The TLRs are just one of those pathways. Many remaining questions, and I'm just posing here two of them for our discussion, is whether we can preserve some of the positive effects of innate immunity on inflammation if we target it for pain, and whether there are distinct roles for mechanoinflammation and cellular inflammation or other aspects of inflammation at different stages and phases of disease or in specific patients. So thank you very much for your attention. And I'd like to credit the people involved in this work, both former postdocs Cheng Zhu and Nisha Sambamurthy, as well as collaborators, and my funding sources.
Okay, thank you very much, Carla. So in the interest of time, if you could stop sharing, and Tamara, if you could start sharing, and please feel free to be using the chat box, but we'll be saving questions for the breakout sessions after the 3 short talks. So next, I'm happy to introduce our second speaker, Dr. Tamara King, who is trained in the areas of psychology and developmental psychobiology, and she's currently an associate professor in physiology at the University of New England, and there she's part of the Center for Excellence in the Neurosciences. She previously worked with an internationally renowned pain research group at the University of Arizona, and her current research is focusing on investigating mechanisms mediating bone and joint pain using rodent models of both cancer-induced bone pain as well as osteoarthritis pain. She has funding through the NIH and through other private sources, and I'm happy to let her start giving her talk to us today.
All right. Thanks, Rachel, and thanks for inviting me to be part of this session. Really excited about it. As indicated, I'm going to be talking about kind of how do we measure pain and how do we think about what that means. And I will be focusing on rodent measures of pain because most of our models are rodent models, but happy to discuss other animals during the breakout of the panel. And so I wanted to give this as a sky-high simplified version of how pain is transmitted. And so in an osteoarthritic joint, as indicated by Dr. Scanzello, there are multiple factors that can contribute to pain. We have the pathology, mechanical contributors, inflammation, that was a lovely overview, as well as some indications that there might be neuropathy or neuropathic pain components to osteoarthritis in subsets of patient populations. These signals are propagated by primary afferent neurons to the spinal cord, where they synapse onto second-order neurons, and the pain signal continues to project to the brain. What's really important to realize here is that it doesn't just project to one nucleus or one area. It projects to multiple brain centers, including the cortex, the limbic systems, and the brainstem. And processing within all of these areas is what gives us the perception of pain, the full experience of both sensory and emotional components of it. Now, these areas can also modulate the pain that we experience. And a lot of work has been done on descending modulation, where descending projections from the brainstem, but other areas projecting to the brainstem, can either enhance your pain through descending facilitation or diminish your pain through descending inhibition. So, as was just indicated, you can have peripheral sensitizers acting on the primary afferent neurons. You can get changes within the spinal cord, resulting in spinal sensitization and signal amplification of the second order sensory input. And then, of course, you can have changes within the brain and descending from the brain that just amplifies this whole thing. So there's a lot to examine, think about, and look at when you're talking about any chronic pain state, including osteoarthritis pain. So, as has already been indicated, osteoarthritis pain is very complicated. What our laboratory kind of does is try to figure out what's different between these different stages of pain. And we really focus on mid-stage OA, where you have predictable pain that becomes more constant over time and is really associated with joint use, starting to interfere with everyday activities such as climbing stairs. I can relate to that quite a bit. Then we have some populations that develop what's called advanced OA, and this is associated with development of constant dull or aching pain state that is resistant to NSAIDs. So it's resistant to most medications. And these patients often start to show avoidance of activities, and they're many times the ones who undergo the joint replacement therapy for treatment of their pain. So when thinking about, well, how do we get at a better understanding of mechanisms driving the pain, as well as, hey, I have a new treatment, is it effective in treating OA pain? We have to think about, well, what is OA pain? As indicated by the multiple mechanisms, there are also multiple outcomes. And so patients describe hypersensitivity to normally non-noxious stimuli, so putting weight or pressure on the joint, as well as movement-related pain can be outcomes. And in a rodent, we can actually kind of measure these fairly decently, especially if we're using a joint that is highly impacted by weight-bearing or movement-related pain. The other kind of pain that patients can describe, spontaneous or ongoing pain, can be a little bit more tricky in the rodent. This is pain that is just there, and in the clinical setting is typically measured using verbal responses, such as rate your pain on the visual analog scale, or describe the characteristics of your pain using a McGill questionnaire, for example. And so how to get at these aspects of pain in the animal has been something that the pain community has been really engaged in and thinking about. And I'll describe a little bit about what we're doing in our lab to get at that question. So the most commonly used measures for a very, very long time had been reflexive withdrawal of usually the hind paw, because that's easy to get to at a rat or mouse, from noxious or non-noxious stimulation. And this was to model things like hyperalgesia and allodynia. Widely used, really, really convenient for things like dose response curves, time of treatment or drug effects, that kind of thing. But people were starting to realize that they don't really translate well into the clinical setting. So they're not very predictive on efficacy of a potential compound to treat a pain state. So people began expanding their arsenal of ways to measure pain. One of the ways that they expanded was to look at things like shifts in weight bearing, guarding behaviors, and different aspects of limb use and gait. And these indeed might reflect enhanced pain due to weight on the joint or ambulation, but they may also reflect other things, such as learned behaviors. If every time you put weight on a joint, it hurts, you're going to learn pretty quickly not to put weight on that joint. And so guarding behaviors or a shift in weight asymmetry might reflect that learning, even in the absence of ongoing pain. Is this still relevant? Yes, but it's not necessarily directly measuring a pain state. And of course, any change in gait or guarding or posturing of a hind limb or any limb may be a result of joint pathology that results in altered posture or gait. Now, these are still really useful tools. They might not be totally getting at only pain, but they are reflecting alterations in the use and function of the joint that are also observed in humans. And so they might have better face validity for translation to the clinic. Now, as I mentioned, we are pretty good at getting at sensory discriminative aspects of pain in the preclinical models. But what's really been sought after, probably across at least the last decade or so, are measures that capture the affective or motivational aspects of pain, the unpleasantness or the urge to escape. And this is really important because it underlies fundamental reasons for pain. Pain is a body defense system, right? So it teaches you things. It teaches you to escape something that might damage you, to avoid such situations in the future. And so we might want to start tapping into those aspects to get at the motivational aspects or the unpleasantness of pain. On the other hand, removal of an aversive state, such as chronic pain, can be rewarding. And this is a concept known as negative reinforcement. And so pain relief is a prime example of this. If you take a medication and your chronic pain goes away, you're more likely to go seek that medication again. And in the clinical setting, pain relief has been reported to feel good. And in the preclinical setting, there are a variety of measures indicating that pain relief can activate the reward pathways, such as inducing dopamine release in the nucleus accumbens. So one of the ways we started to bring these into laboratory is using motivational aspects of pain relief to unmask and to learn about spontaneous or ongoing pain. And so what we did is we adapted condition-place preference from the addiction literature. And here we use a 3-chamber box that you can see have visual cues, textural cues, and odor cues. And what we do is a 3-day procedure. And very, very quickly, what we do is we have 1 day where the animals just explore the boxes across 15 minutes. And we measure the time the animals spend in each of the boxes. And we calibrate the boxes so that the animals spend equivalent amounts of time in each of the pairing chambers, the outside chambers. Second day is called conditioning day. And what we do is in the morning, we give the animals a vehicle and we confine them to one of the chambers for 30 minutes. Take the animal out. 4 hours later, we give the animal a pain alleviating drug and confine it to the opposite chamber. We then take them out, put them in their facilities overnight. The next day, we put them back in the chambers and let them explore. And this is really important. This is in the absence of any manipulation or drug. So we're kind of alleviating some aspects of like motor impairment or some other impairments that the drug might induce. And what we're doing is we're looking at the prediction that rats with injury will spend more time in the chamber paired with the pain alleviating drug. And across so many models now and across multiple laboratories, this is routinely what is observed. Animals in the state of chronic pain will show increased time spent in the pain alleviating drug paired chamber compared to their preconditioning time. And this is, if you subtract these, then you get a positive number. Usually I show these because they're easier. And so this only works if there's an underlying tonic pain state. Otherwise, there's no pain to relieve and nothing to be gained by pairing pain relief with that chamber. And so we have characterized this. We've showed that multiple times that pairing treatment that alleviates chronic pain produces CPP. And that this can be observed by drugs that don't directly activate the reward centers, and that this only occurs in the context of chronic pain. If the animal's not in pain, it doesn't happen. And so relief from the pain-induced aversiveness elicits motivation to seek relief. Now, what I have been doing in our laboratory is kind of combining all of these concepts to try and get at the complexity of different stages of osteoarthritis. And so what we wanted to do was find out what's different between mid-stage and advanced OA. So we use the MIA, monosodium iodoacetate model, where we inject the compound into the knee joint unilaterally. And this reliably in ours and many, many other laboratories produces weight asymmetry, and typically in a concentration-dependent manner. But to get to advanced OA, we really need to know whether we have that constant dull aching pain. And so we turn to our condition-place preference paradigm. We do peripheral nerve block by lidocaine injection into the knee joint. And lo and behold, we can detect different concentrations that produce mid-stage osteoarthritis compared to advanced osteoarthritis. And so this kind of opens things up to where we can look and ask about whether advanced OA develops in other models, such as the surgery-induced osteoarthritis models, and does OA pain correspond to pathology? And we're starting to get an idea that just as in the clinical setting, it doesn't necessarily, pathology does not necessarily predict pain. Females will show ongoing pain, so condition-place preference, in this case the duloxetine, at a 5-fold lower MIA concentration compared to males. And these pathologies, so males treated with this concentration, show equivalent levels of pathology, but very different pain states. And so we're excited about examining that further. So another measurement of pain is alteration of voluntary wheel running, which we have been using to look at movement-associated pain. And this can be observed if the osteoarthritis is induced prior to access to wheels or in a reversal paradigm. And we've shown this, other people have shown this. What's interesting is other people have shown reductions in wheel running in models of chronic pain such as headache. And so it could be that there is a distinct pain component to this, but we acknowledge that it could also be relation of joint pathology to running because we are looking at the knee joint. And I'm not going to spend too much time on this slide because of time. It was already described, but people have been using cage behaviors to assess pain. This is from Rachel Miller and Anne-Marie Malfay, and they looked at various behaviors in the cage and show that a time-dependent emergence and reduction in these cage behaviors. And what was really interesting to me is that these differentiated between tactile hypersensitivity. So you're starting to get the idea that different behaviors can be modulated in different ways, hinting at the complexity of the pain state. And so 3 things to think about is when you're looking at these measures, you need to think really hard about the interpretation and whether you have the proper controls, what's being measured. You really should try and use pain measures that have been validated within the literature for things like pharmacological efficacy and face validity. Things that work in the clinical setting should work on your pain behavior, and it should be appropriate. And preclinical measures should relate to the targeted clinical symptom. That's why I really like these measures, such as the voluntary wheel running and the cage behaviors. And so I will end by acknowledging, of course, my funding, as well as my lab group and my collaborators. We do a lot with looking at the pathology of the joint to see how the pain associates with it. And I will leave off there.
All right, thank you very much, Tamara. Okay, so we'll get to our third talk. And Jason, why don't you go ahead and get your slides going. So our third speaker is going to be Jason Franz he's an associate professor in the UNC Chapel Hill and North Carolina State University Joint Department of Biomedical Engineering. Jason received his bachelor's and master's degrees in engineering mechanics from the University from Virginia Tech, and received his PhD in integrative physiology from the University of Colorado Boulder with Roger Crom. So Jason's expertise is in the neuromechanics of limitations in walking ability and falls among older adults. And more recently, he's received NIH funding to identify techniques to optimize rehabilitation in those at risk of developing OA. And so we're really excited to have Jason here today. This is his first ORS meeting, so welcome, Jason, and we look forward to hearing your talk.
Thanks, Tim. Can I get the mic checks okay and the video's good? Perfect. So thanks for the introduction. As Tim said, this is my first time at ORS, so it's a pleasure to be joining you and the other speakers. I've already learned so much. I admit my talk is going to be somewhat tangentially related to pain and osteoarthritis. I am with the Joint Department of Biomedical Engineering. It's a unique program. It spans both UNC Chapel Hill and NC State. And in the spirit of this research interest group, we'll see if we can't bridge some disciplines, because I feel like that's exactly what we've been doing over the past few years through a relatively new line of research for my lab that we're doing in collaboration with the UNC Motion Science Institute. And that collaboration has been really fun because we've been able to leverage our experience in the biomechanics of aging and some of our rehabilitation engineering infrastructure in brand new ways that we think may contribute to our collective effort to understand and mitigate the onset and progression of osteoarthritis. So researchers at UNC Chapel Hill have long studied ACL injury and subsequent reconstruction as a model for the development of osteoarthritis, specifically post-traumatic osteoarthritis, or PTOA. There's well-documented strength and activation deficits in the quadriceps muscles following ACL reconstruction, which are at least accompanied by altered knee joint biomechanics considered relevant to OA. Now, because the quadriceps muscles are by far the largest contributor to joint contact forces, those changes are then thought to alter cartilage loading patterns to magnitudes and locations not protected by prior loading experience, and with implications for the onset of pain. And there's a lot about this neuromechanical cascade that we don't understand. And I admit, I said we're relatively new to this, but I'd like to focus on what I see are two major gaps in our understanding. The first is that deficits in quadriceps muscle force generation, subsequent changes in gait biomechanics, are not simply governed by muscle weakness and inhibition. There's this black box here that's poorly understood even in healthy controls, and that is the underlying mechanical behavior of the muscles themselves. The second is that most studies trying to associate those altered gait biomechanics with altered cartilage loading have been purely observational in nature, and it's incredibly difficult to establish cause and effect relations that may predispose to osteoarthritis. If we can overcome just some of these challenges, we may be able to identify new or more effective translational opportunities for OA prevention. So the specific tools and experience that we're going to draw on to address these gaps have really come from sort of repurposing our research programs. Specifically today, I'd like to share with you opportunities to use in vivo ultrasound imaging, real-time gait biofeedback, and musculoskeletal modeling to address two key questions. The first is, how does joint injury impact quadriceps muscle mechanics underlying those gait biomechanics relevant to osteoarthritis? And the second is, can we manipulate joint loading and tissue biochemistry to optimize rehabilitation and prevent OA? Perhaps in our discussion that follows, we can work together to find ways to integrate some of our discoveries into the broader conversation of our colleagues on inflammation and pain in osteoarthritis, or applications of our approaches to osteoarthritis more generally. So we know, like I said, that quadriceps dysfunction following ACL reconstruction, it's accompanied by significant reductions in peak knee extensor moment, which you can see here on your left, and knee flexion excursion, which you can see on your right during the early stance phase of walking. And both these changes are evidence of quadriceps avoidance during walking. But it's clearly an incomplete story because while conventional resistance training can actually restore muscle strength, those interventions do little to mitigate these gait changes, which are much more persistent. In vivo ultrasound imaging provides a way to look under the hood to discover how these muscles are actually operating to produce knee moments and the emergent joint contact forces considered so relevant to OA. Specifically, our work focuses on the vastus lateralis, which by its size alone is the largest muscular contributor to knee joint moments and joint contact forces. You first see here in the middle, the length change of the entire muscle tendon unit, which we can derive as a simple function of knee joint angle. And we focus on this shaded weight acceptance phase during which the quadriceps muscles are generating force to actively resist knee flexion and muscle tendon unit lengthening. However, underlying those joint mechanics is actually something really surprising and at odds with the way that we, including myself, have taught about muscle mechanics and its application to walking. And that's shown here. The quadriceps muscles themselves are actually shortening with knee flexion as the joint is being loaded. What that implies is that tendon lengthening, not muscle lengthening, is more responsible than previously appreciated in governing quadriceps muscle forces during limb loading in walking, at least in uninjured controls, which are shown here. So what happens following ACL injury and subsequent reconstruction? Well, let's retake our ultrasound measurements during walking in individuals 6 to 12 months after surgery. And as a reminder, these individuals walk with considerable reductions in the knee extensor moments generated by these muscles. The first thing we're going to see here is that the quadriceps muscle contractile behavior on the uninjured contralateral limb is largely indistinguishable from that in uninjured controls. But we see fundamentally different behavior on the reconstructed limb. Only in that limb do we see significant muscle lengthening during limb loading, something even more surprising considering that this limb is going through much less knee flexion excursion than the uninjured controls. So what does that mean? Well, we have at least two possible explanations, both of which we feel are relevant to strategies seeking to optimize knee joint loading following HCL reconstruction. The first is that due to insufficient muscle force, the quadricep muscles themselves may be either succumbing to the task demands of weight acceptance during early stance, or they may actually shift to eccentric muscle action as a compensatory strategy. And the second is that these individuals may be operating their muscles in a way to avoid excessive tendon lengthening, something especially relevant in these subjects because they all received bone patellar bone grafts. And sort of as a takeaway here, the data suggests that quadriceps behavior may serve as a marker of recovery or at least tendon pain or a target for intervention to reduce the risk of post-traumatic osteoarthritis in individuals following ACL reconstruction. So let's move on to our second key question. Can we manipulate joint loading and tissue biochemistry to prevent osteoarthritis or to have a meaningful impact on pain? So people following ACL reconstruction, they tend to walk in a way that we would expect from the knee extensor moment here to load articular cartilage with magnitudes less than that in uninjured controls. And this idea of underloading has implications for cartilage tissue integrity. For example, in 2018, we showed that individuals with less limb loading, measured here by smaller peak vertical ground reaction forces during early stance, exhibit larger changes in serum cartilage oligomeric matrix protein, or COMP, after just 20 minutes of walking. And this is at least indicative of more cartilage breakdown in individuals who exhibit less limb loading during walking. And this is sort of where we join forces because we've been interested over the last 10 years or so in the use of real-time biofeedback, both as an experimental tool and as a method to enhance or optimize rehabilitation. And in the study here, we set out to determine if augmenting limb loading during early stance in walking using biofeedback could also augment that biochemical response relevant to cartilage tissue integrity. So just briefly, on different days, separated by about a week, subjects walked while watching a video screen showing their instantaneous peak vertical ground reaction force on a step-by-step basis, as well as target lines on different days representing usual loading, lower than usual loading, and then higher than usual loading. And we measure that change in serum comp following 20 minutes of walking with each of those conditions. The key outcome from that study is shown here. So we found that prescribing higher than usual loading values with biofeedback actually decreased the change in serum comp, suggesting that we can augment joint loading and tissue biochemistry with biofeedback. The problem is that peak vertical ground reaction force is only an indirect limb level surrogate for knee joint loading. So can we more directly target the knee? Well, in more recent work, we've been performing more direct real-time estimates of peak knee extensor moments on a step-by-step basis. And you can see here, those estimates agree pretty well with those derived from inverse dynamics. Here we're using visual biofeedback to encourage subjects to walk with larger than normal peak knee extensor moments, which are the dots shown in green, and to walk with smaller than normal peak knee extensor moments, which are the dots shown in blue. And in doing so, you can see here that we can successfully manipulate knee extensor moments to prescribed values shown on the left, and that those changes are accompanied by systematic changes in knee flexion excursion. In other words, subjects are encouraged to overcome that quadriceps avoidance strategy underlying stiff knee gait thought to contribute to altered cartilage loading. But let's not stop there because I think we can do better in terms of establishing cause and effect relations between altered gait biomechanics and cartilage tissue loading. And that's an absolutely ideal opportunity to leverage some of the recent advancements in computational musculoskeletal modeling and simulation. In our case, the pipeline we're going to use is called COMAC. It was developed in part by a friend of mine, Colin Smith, which stands for Concurrent Optimization of Muscle Activations and Kinematics. And there's now an open source version in OpenSim. So this is a concurrent solution which simultaneously solves for muscle forces and joint mechanics. And another benefit here is that we can simulate combinations of muscle activation weights and soft tissue properties to account for functional and anatomical variability. Here we're going to drive these simulations using measured whole body motion capture and bilateral ground reaction forces as subjects walk using this real-time knee extensor moment paradigm, this biofeedback paradigm. And these preliminary data from some of our simulations to date with all the data points presented here relative to normal walking without biofeedback. So as an example of just some of the outcomes that we can derive, on the left, we see change in anterior tibial translation. And on the right, we see the change in peak force transmitted through the ACL. You can see that both of these outcomes are sensitive to prescribed changes in peak knee extensor moment, but that upturn at the lower values suggests this relation may be more complex than we would have previously appreciated. There in the middle, you can also see variation in the ACL force across those 100 different combinations of soft tissue parameters. But what I'd really like to focus on is this, the change in peak cartilage contact forces elicited through biofeedback, which I've separated here into peak force on the medial tibiofemoral plateau as the closed symbols, and on the lateral tibiofemoral plateau as the open symbols. There's really two things I want you to come away with from these data. The first is that this gives us some confidence that we can actually manipulate tibiofemoral cartilage contact forces to larger and smaller than normal values using knee extensor moment biofeedback. And the second is that those forces on the lateral plateau, they appear to be more sensitive to these prescribed changes, which is at least consistent with the larger size and force generating capacity of the vastus lateralis muscle. So it may be accommodating more of the change in knee extensor moment during early stance. Now, right now, it seems a bit ridiculous to think about clinical feasibility and scalability, but I'll at least say that recent advances in machine learning and wearable sensors could eventually be able to overcome those challenges and allow for some scalability. The data suggests that we may be able to use an approach such as this to augment, perhaps optimize mechanical loading at the knee to preserve or restore cartilage tissue integrity in hopes of preventing the development of osteoarthritis. So a few take-home messages really briefly. ACLR reconstruction fundamentally alters quadriceps contractile behavior underlying knee joint biomechanics considered relevant to OA. Quadriceps muscle behavior may serve as a marker of recovery or a target for intervention to reduce the risk of post-traumatic osteoarthritis in individuals following ACL reconstruction. Targeted biofeedback can manipulate knee joint loading and tissue biochemistry with implications for optimizing rehabilitation. And then this last one's sort of really high level. Musculoskeletal modeling and simulation can provide previously inaccessible insight to predict and thereby optimize joint loading responses to interventions and or perhaps the mechanistic origins of pain following osteoarthritis. So I'd love to acknowledge my esteemed colleagues, my students, especially my senior PhD student, Mandy Munch, who's responsible for a lot of this work, as well as our funding sources. And I'd be happy to take questions during the discussion. Thank you.
Okay, thank you so much, Jason. So now, if we could ask our ORS staff members, we'd like to enable our breakout rooms. So then for all of the participants, you can choose which breakout room that you would like to go into. For some small group discussion, each breakout room will have one of the 3 speakers, and we'll do that for about 20 minutes. And then hopefully we'll still have a little bit of time for everyone to come back into the main room once more. Looks like most people are finding their way back here to the main room. So we have 12 minutes left. The idea is if we can kind of give very short summaries so we can each kind of learn what the other breakout rooms we're talking about. Also, if I could ask the ORS staff to post the poll questions, that will help with planning purposes for next year's meeting. Okay, so if you guys want to fill that out while you're still listening to me here on the side. So for the pain breakout room with Dr. Tamara King, people were asking questions concerning how do you adapt some of these different types of behavioral outcome measures to the different models that are increasingly being used in our field. So there are questions surrounding how could you come up with ways of assessing pain in aging animals or in spine models of osteoarthritis. And so Tamara, I don't know if you want to give a very short summary of what we were talking about regarding that.
Sure. So, I mean, I think one of the key points was that not all of these models are characterized as to whether or not ongoing pain actually develops in them. And so that's one point of interest. If you're going to do CPP or an operant model or something, you might want to do a positive control to make sure that your equipment's working and you do detect the ongoing pain with the manipulation or treatment that you're using. And aging is, of course, up and coming. I think it's going to be really huge. One thing that I've seen reported with aged animals is a reduction of movement overall. And so that's something to be thoughtful of and aware of as you're adapting some of these, especially activity-dependent measures.
Tim and Carla, would you like to go next?
Sure. Well, I'll just give the brief overview. We talked about a number of different topics. Some of the ones that came up were the idea of how do we target maybe some of the innate inflammatory, innate immune pathways without getting rid of the beneficial aspects of them. And so this idea that macrophages in particular are a very complex set of immune cells with both kind of resident and infiltrating macrophages. So how do we target those subsets that are part of the pathology? And this brought in kind of this idea of integrating the disciplines back to loading and exercise and trying to understand what are some of the more pathological effects of that mechanoinflammation versus the beneficial effects. And again, it kind of brings back into that complexity of some aspects of loading with the exercise are beneficial, but obviously other aspects are more pathological, and we don't really have good tools yet to differentiate between those, especially as we try to think more clinically. And then some of the other topics that came up were more sex-dependent mechanisms of some of these inflammatory pathways, such as the TLRs, as well as a role that was just brought up about IL-17 playing a role in cell senescence. So Carla, anything else you want to bring up?
I think you covered it. I just want to expand on the discussion we had about the interplay between mechanics and inflammation, and how one of the potential opportunities that we really need to learn about is how mechanical stimulation of TLR expressing mechanically active cells, the impact of mechanics on these pattern recognition receptor systems that are mostly studied in the context of leukocytes, but there's opportunities to study whether there's an interaction with mechanical input.
Thank you.
Okay, Lou and Jason.
Yeah, sure. We had a very lively discussion in our little group. It was great fun. I definitely learned a lot. We discussed a little bit about, there were some questions related to PTOA and imaging, in particular focusing on the alignment of the joint and how the alignment of the joint may influence how load is transferred through the joint. And we talked about how computational modeling may be able to look at different variables and investigate these kinds of questions. We also talked about whether some of the results that we talked about with regard to ACL injury is whether increasing quad force could potentially reduce OA and pain. And we talked about whether it's potential, there's potentially ways to manipulate the joint loading such that we can find the right amount of loading to minimize pain and maybe minimize OA. And we also talked about some of the decreased loading on the OA reconstructed joints. Maybe that is part of a pain response. And we also talked a little bit about maybe it's possible to make ACL reconstruction obsolete, with the appropriate, whether we talked about copers and non-copers. That was a crazy person who raised that question. So that was me, for the record. When we talked about ways to, maybe we need, using these tools like biofeedback, maybe a way to get around some observational studies that are prevalent in the literature. Maybe we can dig down specific mechanisms, but surely there are other ways we could think about that too. Oh yeah, we talked a little bit about what it would take to get these different gait modifications to be useful clinically. And, could it be used to minimize painful situations? And whether there is a sort of learning effect, once you get out of the lab and you don't have the feedback, can you maintain that outside of the lab? Jason, would you please correct?
That was a really, really good summary. And for the record, I was not advocating for all the orthopedic surgeons here to lose their professions. I thought, yeah, I will say, as a person that has not been to ORS before, I was just fully engaged in learning so much during our discussion and by the other speakers. And the only thing that I wanted to add is, I pitched biofeedback as a potential rehab approach, but I think I'm almost more enthusiastic about it as an experimental tool of any variable. When so many things happen at the same time, it can be really difficult to extract that cause and effect relation. And we've used biofeedback a lot just to take control of a dial and manipulate that one dial and see how it affects other things. And if you can think about ways to do that in your own research, maybe it's not biofeedback, maybe it's other things, but just, I think generally, especially in biomechanics, there's a need to move beyond observational study. And this is just one potential tool that you have at your disposal.
Okay, we can open it up. We have 5 minutes left. So if anyone has any other comments they'd like to add, you can do so in the chat. I'd also just like to mention, especially to the young investigators in the audience, that we're all very welcoming. So if you have questions about how to start studying pain preclinically or clinically, please reach out to us and we'll put you in the right direction. If we're not the right person, we'll put you in touch with the right person. Okay, so I think people like me might be wondering about how to battle the snowstorm to get home tonight. So I'll let everybody start signing off and hope you enjoy the rest of the ORS meeting. And thanks again to the ORS staff for making this happen.
All right, thank you, Rachel. Yes, thanks all the speakers.
Yeah, thanks, Rachel and Tim.
Thank you, Rachel and Tim.
Thanks, Rachel and Tim.
Thanks.
Bye, everybody.